Today is March 25th, 2016. My name is Gregory Williams and I am in Sunrise Beach, Missouri, interviewing Dr. Charles Williams for the Society's Living History Project. Dr. Williams has been a member of the APS since 1983. He was affiliated with Texas Tech University before retiring to Sunrise Beach. Dr. Williams' research focused on an anesthesia problem observed around Wausau, Wisconsin, in both human and pigs, which resulted in high temperatures in swine populations and an adverse reaction to anesthesia in the human population. The topic of malignant hyperthermia became the focus of his research career. Dr. Williams, welcome to the Living History Project, and thank you for agreeing to be interviewed for this series. If you're ready, I'd like to ask you a few questions about your career.
Go right ahead, I'm ready.
If you don't mind, please tell us about your family, your parents' professions, and the number of siblings in your family. Where did you grow up?
I grew up in Ozark Township in Berry County, Missouri, which is located 3 miles south of Aurora, Missouri, which is in Lawrence County. We were right on the county line.
And what were your parents' professions?
My dad was a storekeeper. He had worked for 20 years at the Missouri Farmers Exchange in Aurora, and then in 1939 he opened his own store 3 miles south of town where it was called Williams Produce, and he sold groceries, feed, livestock supplies, and had a 2-pump gas station with it.
All right, and your mother's profession?
My mother's profession was being a housewife and taking care of 11 living children.
Okay. And can you tell us how you became interested in science?
Whenever I started to Aurora High School in 1949, I enrolled in the vocational agriculture classes, and I spent 4 years in those classes. Each year I was tagged by the teacher to do a FFA judging team or competition at the Springfield level and at the state level in Columbia, Missouri. So I did that for 4 years. The first year was field crops, then I did farm shop work, and in my senior year I did livestock judging, both dairy and beef cattle.
And how did that lead you to the University of Missouri in Columbia?
I was well acquainted with the university because I had been up there 4 years in a row, and in my senior year at high school I decided I wanted to go to the university and get a university degree.
And what were your interests when you first attended the University of Missouri?
I started out in dairy husbandry because that's what we had on the farm in Berry County was dairy cows. However, after being there 2 years, I decided I would pursue a degree in vocational agriculture so that I could teach agriculture, not necessarily back in my hometown, but at the high school level.
And you received a bachelor's degree from the University of Missouri. What was that in?
I had a bachelor's degree in agriculture, and I had a specialty of vocational agriculture, which was a teaching degree.
All right, and then from 1958 to 1962, you served in the U.S. Air Force. Can you tell us about that experience?
When I graduated from the University of Missouri, I had completed 2 years of advanced ROTC training, and that made me eligible to be commissioned as a second lieutenant in the U.S. Air Force. So when I graduated in June of 1957, I was awarded a second lieutenant's commission, and I was also given a distinguished Air Force ROTC cadet rating, which allowed me to apply for regular Air Force officer status. When I graduated, I was given a reserve commission. I could convert that to a regular Air Force commission anytime that I wanted to.
And I take it then that you did?
No, I did not. I stayed a reserve officer for the 4 years that I was in the Air Force. I was assigned to Edwards Air Force Base, California as my first assignment. My second assignment was to Hickam Air Force Base, Hawaii, And then my third location was at Wheeler Air Force Base, Hawaii. And by that point in time, I had completed 4 years and 2 months of service, and I elected to get out of the Air Force rather than making it a career.
Okay. What motivated you to pursue a graduate degree after you were discharged from the Air Force?
I had made up my mind during my second year of college that I wanted to pursue a graduate degree and get a PhD. And so the Air Force was a very temporary situation that I felt obligated to do as part of my military service for the U.S. Air Force. And then after I got out, I went back to teaching for 1 year in Vandalia, Illinois. And then while I was there, I made arrangements to return to the University of Missouri and do graduate work in the Department of Agricultural Chemistry.
And can you tell us what your doctoral research focus was at the University of Missouri-Columbia?
My doctoral research was the interaction between estrogen and progesterone on the uterus and how those two hormones could stimulate growth and development of the uterus in order to sustain the embryos that were being developed in the uterus. So it was a kind of a study of the mechanism of cell growth and the development of the uterus into a functional organ which would support embryos.
Now, after you obtained your doctorate degree, you had postdoctoral fellowships at the University of Missouri and also at the University of Wisconsin. Could you tell us about your experiences in both programs and about the people with whom you worked?
My first postdoctoral was in 1968. I moved from the University of Missouri to the University of Wisconsin and joined Dr. David E. Green, who was at the Institute for Enzyme Research as a postdoctoral fellow. And there I was exposed to a lot of research on mitochondria, mitochondrial function, mitochondrial ultrastructure, and I was the first person to actually fix mitochondria in situ in heart tissue and show that there was an energized state where they were twisted and had a very energized appearance in the electron microscopy work.
Okay. And what other experiences did you have at the University of Wisconsin?
Dr. Claude Taylor from the University of Wisconsin Hospital came over to the Enzyme Institute and made me aware of a problem called malignant hyperthermia that was occurring around Wausau, Wisconsin. And this was evident in patients who were getting anesthesia during surgical procedures. They would develop a high temperature and a high heart rate and high blood pressure, and usually the cases were very fatal. There was no way to stop it or reverse it, and as a result, the patients would die. And shortly after, this was in October of 1968, and shortly after he came over, within 2 or 3 weeks after he came over to talk to me, they had a case down at the University of Wisconsin Hospital with a 28-year-old lady who had had 3 previous childbirths with no problem. However, she went in for a thyroidectomy because she had a large goiter on her neck, and this was cosmetic surgery. They gave her anesthesia, they did the surgery, and as the surgeon was finishing up, they noticed that her heartbeat was erratic. They checked her temperature and she was up to 108. They did everything they knew to do at that point in time to reverse the situation, and she died after a matter of, I don't know, 18 to 24 hours. She had already had a hyperthermic episode from the anesthesia she had been given, and they were not able to correct all the problems she had, so she died.
And how did that lead you into your research on malignant hyperthermia?
Well, we were talking about this case down at the hospital. That was a topic of conversation for several weeks. at the Enzyme Institute and at the anesthesia department down there at the hospital. And one of the residents, Tim Galvez, spoke up and said he had been giving anesthesia to pigs for a surgery project, and the pigs would get hot and die before the surgeon ever touched them with a knife. And so he was given a lot of rhubarb treatment because he was a poor anesthetic. He was giving poor anesthetic. Well, it turns out those were the pigs that were genetically susceptible to malignant hyperthermia. So Tim and I went out to the swine farm a few weeks later. I think this was like in November of 1968, and they had isolated one small pig there for us to check. It had a large hernia on the side. It's kind of a throwaway animal. It wasn't going to be turned into pork and bacon and that type of thing. And it was no good for a breeding stock. So they gave it to us to check, and we started halothane anesthesia. We had a rectal probe in to monitor the temperature, and within a few minutes, the back legs on the pig straightened out and went into kind of a rigor mortis-like position, and the temperature went from 97, 98 over 108. That was at the top end of the temperature range. I mean, it went off scale as far as we could tell. And the pig died. And we said, okay, this is the type of animal we're looking for experimental work. And so the animal science people went back into the pig herd out there, and they found relatives of that particular pig that we had checked. And in January of 1969, we went back out to the swine farm, and they had 2 litters of close relatives to that pig, and we checked those pigs with halothane anesthesia. We could give the pig about a 3% halothane anesthetic dose for a few minutes, and if he showed this rear leg reaction and what we thought was a positive response to malignant hyperthermia, we would stop the anesthetic and try to recover the pig. Well, fortunately, we recovered every pig we tested, and I set aside 5 pigs, 2 males and 3 females from that study, from that evaluation, and said, I'm going to save these back for breeding stock. We didn't know what the genetics was. We had no idea how it was transmitted, but I wanted to start a pig colony so we could use animals for research work.
And that enabled you to develop an animal model to mirror the human condition of malignant hyperthermia.
Yes, these pigs turned out to be a very, very excellent animal model for malignant hyperthermia in humans. And I used that same strain of pigs for over 20 years. We raised several litters of pigs over the years, and we found out that this was transmitted as a dominant gene. And through very careful selection of matings and breeding, I was able to produce pigs that could react to halothane anesthesia in 1, 2, or 3 minutes. So this gave us an animal model for the human syndrome.
Can you tell us what type of work you then did with your animal model that contributed to the resolution or a method to deal with malignant hyperthermia?
Well, I read all the literature that had been written on malignant hyperthermia. There were a number of papers from around the world. So I got copies of each one of those papers. I read them very carefully, and it appeared to me from my reading of the case reports that succinylcholine, which is a muscle relaxant, was involved in almost all of the cases of malignant hyperthermia. So I said, okay, let's study succinylcholine in these pigs and see how it reacts in them. And what we found was, if you gave succinylcholine to one of these pigs that was genetically susceptible, it would trigger off a malignant hyperthermia episode that would run to a very high temperature very quickly. So succinylcholine was the first drug that I could point to and say, here is a triggering agent. During that the studies that I was doing, a group in South Africa came up with the idea that you could use dantrolene to reverse malignant hyperthermia. And we tried that, and it worked about 75% of the time. It was not a totally protective agent. So then I pursued the idea that this was a mitochondrial problem. The heat was being produced by an uncoupling process in the pig that would produce heat through an uncoupling process at the mitochondrial level. And I isolated mitochondria from heart, liver, and skeletal muscle. And we studied those in vitro, and we could not find anything wrong with the mitochondria. They were perfectly functional. They did what they were supposed to do. They produced ATP, they consumed oxygen, and they were totally under metabolic control. There was no uncoupling in the mitochondria. So then I got to talking to some of the people upstairs in Dr. Lardy's laboratory, and they were working on a problem in bumblebees. Bumblebees can produce heat on a cold day, and this allows them to fly around and collect nectar when other insects are immobilized. And Mike Clark and some others in Lardy's lab showed that there was a heat generating mechanism at the glucose 6-phosphate level which would generate heat. So we evaluated that in the malignant hyperthermia pigs. What we found was there was a small amount of heat generated by that mechanism in the malignant hyperthermia pigs, but there was not enough to account for the total quantity of heat that was produced to produce the malignant hyperthermia episode.
Okay, and what other testing did you do then with your animal model?
What now?
What other testing did you do with your animal model?
I evaluated a new anesthetic for a Japanese company, Maruyushi of Osaka, Japan. They had acquired a new anesthetic agent called Sevoflurane, and they asked me to evaluate that and see how it performed in the malignant hyperthermia pig. And so I did a year study on that drug, and I was able to show that Sevoflurane was a very good anesthetic. You took about 4 breaths and you were sound asleep. And it did not trigger malignant hyperthermia until it had been on board for maybe 2, 3, or 4 hours. I called it a very slow triggering agent. Therefore, it made it quite safe to use in patients who might be malignant hyperthermia. At that point in time, which was the early 80s, there were no tests for malignant hyperthermia. So these cases could come into the surgery suite, and the surgeons and the anesthesiologists would evaluate them from a personal history standpoint. And if the patient had a personal history, they could kind of identify those that were quite possibly susceptible to malignant hyperthermia. But there was no test you could use that would identify those people accurately.
And Sevoflurane is now the most commonly used anesthetic for that reason, is that correct?
It turned out that Sevoflurane was a very smooth anesthetic, and it was first tested in the Japanese people because Marushi Pharmaceutical was located in Osaka, Japan. Then they came to Abbott Laboratories in Chicago and said, Abbott, we'd like for you to sell this for us in the United States. We don't want to set up a sales force to sell one single anesthetic. So Abbott agreed to sell it, and they had to do another human study in the U.S. in order to get past the U.S. FDA. And that allowed, once that was completed in 92 or 93, that allowed Abbott to put Sevoflurane on the market. And in the year 2000, Abbott was selling $200 million a year of Sevoflurane for anesthesia purposes. It turned out to be a terrific anesthetic for people. It's especially good in young children because it does not irritate the throat. You can put a nose cone over the nose. They can take about 4 breaths and they're sound asleep. And it keeps them asleep during the operation. And when they take away the Sevoflurane, they wake up in about 10 or 15 minutes, and it's just like they've been asleep for a while. It's a very smooth anesthetic.
In your view, what has been your most significant contribution to physiology?
Well, I think my results on the malignant hyperthermia pigs is going to point the way to study heat generation in muscle. We all generate heat in our muscles in order to maintain our body temperature. However, malignant hyperthermia is a runaway situation where the heat is generated at high rates and it raises the body temperature. I had one pig that went up to 118 degrees Fahrenheit. And if you get over 106, you are in the area of where you get brain damage and other organ damage, and the chances of recovering from malignant hyperthermia are very poor.
Tell us about your first APS meeting and how it differed from the current meeting.
My first APS meeting was in Atlantic City, New Jersey. And I went to that meeting to present an abstract, a paper, on my work with the energized twisted electron microscopy pictures that I had developed during my first few months at the Enzyme Institute in Wisconsin. It was on the boardwalk area. It was a very pleasant time from the standpoint of the weather. It's usually in April, and as a result, it was a kind of a pleasant meeting. I really enjoyed it. Now the meetings have been in Chicago, they've been in San Diego. I understand next year they'll probably be in San Francisco. Those are all nice places to go. And I've enjoyed every one that I went to over the years.
What role has APS or other professional societies played in your career?
In 1985, I was able to hold a malignant hyperthermia session at the fall meeting for APS at Niagara Falls. This allowed me to gather about a dozen people from around the world to present papers on malignant hyperthermia, and out of that came a small book called Experimental Malignant Hyperthermia, which we published a year or two later.
Okay. And what advice would you give to students starting out in science today?
For a student starting out today, he needs to take all of the mathematics and all the biology and the hard sciences that he can get either in high school or very early in his college career, because everything in science is really mathematically based, and all the analysis has a mathematical basis for analyzing data and presenting it.
All right, well, that will conclude our interview today. I want to thank you for your time and appreciate your efforts today.
Thank you very much. I've enjoyed the interview.